Question 1 reads, a researcher adds a DNA molecule to a solution that dissolves only sugar molecules. This is going to be important in a moment, that it was sugar molecules. After a time, what would the DNA molecule look like? We need a drawing and a description, and why. Use the words sugar, phosphate, covalent bond, and nucleotide in your answer. Okay, so we're going to start by looking at what the DNA molecule normally looks like. And we're going to start with the sides of the ladder, which are alternating sugars and phosphates. So I'm going to put P's and S's down the side of the molecule. Okay, so if this side over here starts with a phosphate, the other side needs to start with a sugar. And alternate sugar, phosphate, sugar, phosphate. So it's the opposite of the other side because it's in the 5' to 3' direction. All right, so that's the sides of our ladder. The bonds that hold these sugars and phosphates together, those are called covalent bonds. Those are the strong bonds that are hard to break. The rungs of the ladder, or the middle of the ladder, are connected between the sugar molecules. So those sugar molecules have two bases attached between them. So it could be an A and a T, it could be a G and a C, or it could be in the other order, C and then G. But those bases between them are held together with a different type of bond. So between these bases, we've got a second type of bond that's called a hydrogen bond. So two different types of bonds, depending on if you're talking about the sides of the DNA or the rungs of the DNA. The last thing you need to know about is a nucleotide. A nucleotide is always a sugar, a phosphate, and a base. So in this case, the base is a C for cysteine, and the sugar and the S are represented by the S and the P. Here's another another nucleotide with the base guanine. Here's another nucleotide with the base adenine. So all of these nucleotides bond together through those hydrogen bonds to stick to each other on opposite sides, and then through the covalent bonds to stick together on the edges. So there's our basic DNA molecule. Now let's go back and look at what happens if we dissolve the sugar molecules. So if we dissolve sugar, I'm just going to take away some of my markings here. That means that we are, keep going, stay right there. Okay, so if we're dissolving sugar, that means the sugar is going away. So if we imagine all these sugars disappearing, okay, what does that mean for our DNA molecule? Well, that means that our A's and T's, for example, and our G's and C's are still bonded together. Okay, so those hydrogen bonds are still holding the base pairs together, but the base pairs aren't connected to any sugars anymore. Also, you'll notice that our covalent bonds, okay, all of these here, don't have anything to bond to anymore either. So the result of the sugar being gone is that we no longer have a nucleotide. All we have is our bases bonded together. Remember, the bases bonded together are these things, because those are a hydrogen bond holding those together. The sugar is not involved in the hydrogen bond. And we no longer have covalent bonds because the sugar was covalently bonded to the phosphate, so the phosphates are off floating around on their own. So in the end, what it would look like, these little, combinations of G's and C's, C's and T, or G's and C's, C's and G's, kind of floating around, okay, without being connected to any sugars, and then some phosphates kind of floating off on their own as well, because they are no longer connected to the nucleotide. So that's the result of dissolving the sugar.